Hello and welcome to this Carbon Academy tutorial on AQA A-level biology on carbohydrates and lipids. The aims of these tutorials are to give clear and concise information, and all information given here are directly linked to the AQA specification. The words marked in bold are the mark scheme words that examiners look for, and all information given in this tutorial are as accurate as possible. If any errors are found, please notify in the comment section below so it can be corrected as soon as possible. And these are the AQA specification points. I'm not going to go through them in a much great detail, um, but you may feel to make a note of them. So let's start with monomers and polymers. Mono means one and poly means many. And these polymers are made out of many units of the monomers. These monomers make these polymers via a condensation reaction, and the polymers can be broken down into monomers by hydrolysis reactions. Proteins or polypeptides and carbohydrates or polysaccharides are both polymers that you need to know for AQA A-level biology. And this table summarizes the associated polymers and monomers and the bonds holding them together. In this lesson, we will not be going through polypeptides, only polysaccharides and lipids. The monosaccharides you need to know for A-level biology are glucose, fructose, and galactose. Condensation and hydrolysis reactions. Let's see this in more detail. So a condensation reaction enables for a larger molecule to form, and a water molecule is released in this reaction. And in an hydrolysis reaction, bonds are broken to form smaller molecules, and a water molecule is added in these reactions. For example, here are two alpha glucose molecules, and these two alpha glucose molecules make the disaccharide maltose. Disaccharide meaning made up of two monosaccharides, and here they form a 1,4 glycosidic bond. There's the carbon 1 on this side here and the 4 carbon on this side, so 1,4 glycosidic bond. The water is made by the hydroxyl group on one of the alpha glucose and the hydrogen group on the other, and via a condensation reaction this disaccharide is made, and this disaccharide can be broken down to the monosaccharide via hydrolysis. So hydrolysis means breakdown with water, so hydro meaning water and lysis meaning breakdown, so hence the reason why you can see larger molecules can be broken down into smaller ones. Formation of disaccharides. As I told initially, disaccharides are made out of two monosaccharides via a condensation reaction. Monosaccharides include glucose, fructose, and galactose, as I mentioned earlier. Glucose has two isomers, alpha and beta. I'll be discussing this in the next slide. The monosaccharides listed are all hexose sugars, meaning they contain 6 carbons in their structures. And the table below is showing the disaccharides that you must know and the monosaccharides that make them. So isomers of glucose, isomers mean they have the same formula, so C6H12O6, but the structural arrangement is slightly different. As you see in the alpha glucose, the hydrogen groups on carbon 1 and carbon 4 are on the same side, whereas for the beta glucose, it's inverted, so the glycosidic bonds on beta glucose are inverted. And alpha glucose is used to make the polymers such as starch and glycogen, whereas beta glucose is used to make the polymer cellulose. So there are two types of sugars depending on their reducing abilities. They're called reducing and non-reducing sugars respectively. And reducing agents are electron donors. They themselves are oxidized. All monosaccharides and most disaccharides are reducing, while all polysaccharides are non-reducing. And sucrose is the most common non-reducing disaccharide. So the tests for these types of sugars are as follows. Heat the sample with a Benedict solution in a boiling water bath. and if a brick red precipitate forms, there's a reducing sugar. And add a Benedict solution to make sure all sugar has reacted. This third point is not often asked in exam questions, but for a three-marker question, it is best to put this as the third point. For non-reducing sugar, you have to carry out additional steps, and these are the steps. Heat the sample with a dilute hydrochloric acid in a boiling water bath, and this will hydrolyze the sugar, breaking the glycosidic bonds, and the reducing sugar made is a monosaccharide. Add sodium hydrogen carbonate, a alkali, to neutralize this acid in order for the Benedict solution test to work, and carry out a normal Benedict's test, and if there is no color change, there is no non-reducing sugar present, so there is no sugar at all. And this diagram summarizes the precipitate that you are wanting to see. So the greater the amount of the reducing sugar, the greater change in the color of the precipitate. So let's have a look at the polysaccharide glycogen into more detail. It is made of alpha glucose, and it is a very efficient source of energy as it has many branches and is very compact. It is also insoluble, so it doesn't affect the water potential, so it can be stored without affecting the cell volume. And because of its branches, it can be rapidly hydrolyzed into glucose and can be used for respiration. And here is the structure of glycogen. As you see, there are many branches. It allows enzymes to break those glycosidic bonds and any other bonds that in between, and to release glucose quickly for respiration. Let's have a look at the polysaccharide cellulose. It is a long unbranched chain of beta-glucose joined together by glycosidic bonds. The hydrogen bonds are actually between the layers of the chains of beta-glucose. The hydrogen bonds allows strong microfibrils to form, which provides strength and rigidity for plant cell walls, and it helps to maintain turgor pressure in the plant cells. So the turgor pressure is the pressure created by water inside the plant cells, and because these cell walls do not burst when there is a high turgor pressure, it allows the plant cell to stand upright. If there is less water, such as in a dry condition, the plant cells would wilt as there is less water and less turgor pressure generated. Here is the structure of cellulose. Again, it has a 1,4 glycosidic bond. The light blue dotted lines indicate the hydrogen bond formation. The hydrogen bonds form because of the electronegativity of the oxygen, and the electronegativity of the oxygen attracting the partially positive dipole of the hydrogen from another layer of the cellulose. This forms a strong microfibril. This is the polysaccharide starch, which consists of amylose and amylopectin. Amylose is helical and unbranched, so it is compact. And amylopectin is long and branched because it has 1,6 glycosidic bonds between the glucose molecules, and this allows the enzyme amylase to reach and hydrolyze these glycosidic bonds quickly, releasing the glucose rapidly for things such as respiration and other reactions. This is the structure of amylose. It is, as you see, it is helical, allowing it to be compact. And amylopectin, which consists of branches, clearly different from amylose. Amylopectin has a similar structure to glycogen as we saw earlier. So starch is a very efficient storage molecule because it is insoluble and it does not affect water potential. It is large and does not cross the plasma membrane. And for a starch test, you add iodine dissolved in potassium iodide. And if the color changes from brown to black, there is starch present. It's a common test that you would have done in your years earlier. And this is the image indicating whether starch is present or not. So lipids are not polymers. Lipids consist of triglycerides and phospholipids. Triglycerides consist of three fatty acids attached to a glycerol molecule by ester bonds, and these fatty acids are considered hydrophobic, hydro meaning water, phobic meaning fear. So they're not fear of water, they repel water, they do not dissolve in water. Lipids do not dissolve in water. Phospholipids contain two fatty acids, a glycerol molecule and a hydrophilic phosphate, and these fatty acids can be saturated or unsaturated, depending on their carbon-to-carbon double bond. Saturated fatty acids have no carbon-to-carbon double bond, while unsaturated fatty acids do have a carbon-to-carbon double bond. And this is the structure of a phospholipid. You do not need to know the full structure here. It is just for understanding purposes. The phosphate head is considered hydrophilic due to its negative charge, so it can dissolve in water. Hydro meaning water, philic meaning loving, so water loving, so it dissolves in water. And the fatty acids are hydrophobic. This allows the phospholipids to form the plasma membrane, the bilayer, which I'll be discussing in the next few lessons. So properties of triglycerides include they are a long-term source of energy, they are hydrophobic, so they repel water, don't dissolve. Hydrophobic and non-polar are interchangeable terms. They do not affect the water potential, so it can be stored. And because the fatty acid tails are long, a lot of energy can be stored. And the large molecules do not cross the plasma membrane, hence enabling them to be stored easily. This image illustrates how triglycerides are made. Again, this is a condensation reaction as water is released, and the ester bond highlighted here indicates how the bond looks. So there are three fatty acids attached to this glycerol molecule. For those who are chemists, you would need to learn this structure of the glycerol. For the non-chemists, you do not need to worry about it, i.e. biologists. For the lipid test, you can test whether a substance contains lipids by shaking it in ethanol and then adding water. So then is a key word here. You do need to add water after adding ethanol. And if a milky white emulsion forms, lipids are present. And the more the lipids, the more noticeable the white emulsion. And here is a test that indicates whether lipids are present or not. That is the end of this tutorial. Thank you for watching, and I'll see you in the next tutorial.